Hello, you're welcome to Medical Sciences Made Easy by Naphtali Muhumuza. And in today's session, we want to look at DNA replication, which is part of protein synthesis. We know protein synthesis, or what we call the central dogma, is made up of what we call the central dogma. Is that when we have the DNA, this DNA undergoes replication to form an mRNA. It undergoes replication, and then this replication is replicated, that is duplication, to form copy, that is replication. After being replicated, this replicated DNA, or the daughter DNA formed from the daughter DNA, undergoes transcription to form mRNA, and this mRNA is translated. It undergoes translation to form protein. And this is what we call the central dogma of DNA, whereby we are copying information from the DNA, we translate it into a single-stranded RNA, which is going to use as an adapter in the presence of ribosome to transform it into a protein. So this is what we call the central dogma, which is, in the central dogma, we have processes like replication, transcription, and translation. So in today's video, we want to focus on DNA replication, whereby DNA replication is duplicating. Duplicating means that if I have the parental DNA, if this is the parental DNA, we are going to see this parental DNA being duplicated into two, whereby we form two daughter DNAs, each having one template of the parent DNA, whereby this has one template of the parent DNA, and this has a template, and then it has also another newly synthesized strand. And this one also has another newly synthesized strand. So these ones we form two daughter DNA using a one parent. This is a parent DNA which have undergone duplication to form two daughter DNA, each having one strand from the parent. And this theory where it works is what we call semi-conservative, which was this semi-conservative theory was described by Crick and Watson, that during DNA replication, we see each daughter strand, each daughter DNA taking up one strand from the parent and another one which is newly synthesized. And this is what we call semi-conservative theory under DNA replication. And this DNA replication, we are going to see the steps involved and the enzymes that we shall be talking about involved in DNA replication. We have what we call DNA-dependent DNA polymerase. It is DNA-dependent DNA polymerase, and in prokaryotes, it is named in Roman numerals. This is in prokaryotes, whereby it is DNA polymerase type 1, DNA polymerase 2, and DNA polymerase 3. This is in prokaryotes. Then in eukaryotes, it is named according to Greek letters, and this is whereby 1 is alpha, then 2 matches together with beta and epsilon plus gamma, whereas 3 is the same, has the same function with the delta in prokaryotes. So this is the major difference of these DNA-dependent DNA polymerases. Whereby this is moving with the delta, and the 2 is moving with the beta, epsilon, and gamma, then 1 moves with alpha. Prokaryotes in Roman numerals, then eukaryotes in Greek letters. And we shall see their function as we go on in the process of DNA replication. So the enzymes involved is this, the major one, and we shall see other enzymes like helicase enzyme. Then we shall see DNA gyrase also doing the role in this process of replication. This one you can call it topoisomerase. Topoisomerase, which we call DNA gyrase, and we shall also see another substance another enzyme known as plymase, and this is called RNA plymase. This is plymase enzyme. It's called RNA plymase. Then we shall see others like other proteins like DNA protein. Then the helicase is DNA B protein. Then finally we shall see single-stranded binding protein also doing the role. So these are the components that are involved in DNA replication, and we shall see that they are involved in steps of initiation, elongation, and termination. We are saying DNA replication occurs in 3 stages. Initiation, the second one is elongation, the third one is termination. And these processes in the DNA, we are going to see the interplay of these enzymes. DNA polymerase, helicase, DNA gyrase, primase, how they are bringing about this process. And we want to start with initiation step. Step number 1 is initiation. And this initiation is that before the DNA starts to be replicated, if this is my DNA, parent DNA, before replication we need to first recognize the point of origin, or what you call origin, site of origin. And what recognizes, so let us say that the site of origin of replication is this one. We are going to see a protein known as DNA A protein, which comes and recognizes the site. Because we see in prokaryotes, majorly we are going to see this replication in prokaryotes, whereby it has a specific recognition site, or they have a specific site where replication starts. And it is recognized by DNA A protein, and this protein is the one that recognizes the origin of replication, where the replication fork starts to be formed. So after recognizing the origin or the site, it denatures the strands. And after denaturing the strands, it creates a way for helicase to come in. So it comes and denatures these strands, whereby it unleashes them, and after opening them, then DNA helicase comes and sits at the replication fork. So we are going to see that what initiates unwinding by DNA helicase is DNA protein. DNA A protein. It is the one that recognizes the site. And we see in prokaryotes, they have a single specific site. Whereas in eukaryotes, they have multiple sites of origin. So in prokaryotes, they have only a single site of origin, which is recognized by DNA type A polymerase, what we call the DNA A protein, which is recognizing the origin. And when it recognizes, it binds and denatures the strands, allowing or paving the way for helicase to come and start cutting. So we are seeing this initiation step. We needed DNA A protein, which has recognized the specific site. We said in the prokaryotes they have single, then in eukaryotes they have multiple sites. And it is the one that paves the way for helicase to enter and start unwinding. So this one is called the helicase enzyme. This is helicase or you can call it DNA B protein, which comes after the denaturation of the strands by DNA A protein. Helicase comes at the point of origin and he comes with a molecular scissor, and he starts cutting the strands and winding the DNA strands. So we see he starts cutting and winding or separating the two DNA strands, and as he's separating them, he causes the stress. So what cuts or unwinds the strands is DNA helicase. So helicase is the one responsible for unwinding of the DNA strand. It unwinds DNA strands. So it is the one that separates the double-stranded DNA into separate RNA strands. And after separating these strands to make and at the point where it is cutting is what we call replication fork. So he continues cutting forward. He continues cutting, and this is the direction of replication fork. And as he continues to cut, he will create a pressure in front. And this is that if this is my DNA, we are going to see the DNA type DNA protein denaturing the strand to pave the way for helicase. So helicase comes and stands here and begins cutting the DNA strands. But as he's cutting, he creates pressure in front. It reaches a point whereby we need a bulldozer, whereby at this point, due to the stress created by helicase, it can no longer cut to unwind the DNA strands. So because it cannot unwind, we need a powerful enzyme. And this powerful enzyme is known as DNA gyrase, or what we call topoisomerase. So it is topoisomerase that unwinds the supercoils created by helicase enzyme. That as helicase is cutting, he creates stress, causing the strands of DNA to become tight, and he cannot manage it to continue. So he calls topoisomerase, or what we call gyrase, to unwind the supercoils so that the helicase can continue doing the role. So we are going to see that as helicase is cutting, at this point he creates what we call supercoils. These are what we call supercoils. And these supercoils, helicase has no energy to unwind them. That's why we see an enzyme which is known as DNA gyrase. This is topoisomerase comes in. You can call it topoisomerase or DNA gyrase, which is acting as, it has more energy than the helicase. So he's the one that uncoils the supercoils to pave the way for helicase to continue unwinding. So topoisomerase does not unwind. His role is to loosen the supercoils created by helicase as he is cutting. That is the function of topoisomerase. And we shall see certain drugs which target this, like ciprofluoroquinolones, like ciprofloxacin, targeting this DNA topoisomerase, that whenever helicase is cutting and creates supercoils, there is no gyrase to break the supercoils, and hence replication cannot occur. So then the DNA or the bacteria will not multiply. And after saying that, we are going to see now the synthesis. That is the initiation step. So under initiation, we have seen the action of DNA A protein, which recognizes the origin. It binds at the site at the specific site, recognize the specific site, and creates origin of replication. And after doing this, it denatures the strands. After denaturing the strand, it was paving a way for the action of DNA B protein, which we have called helicase. And it is this helicase enzyme that is responsible for unwinding or separating the DNA strands. So he separates the DNA strands, and after separating, and as he's cutting, he creates supercoils, and we have seen these supercoils are removed by topoisomerase. Topoisomerase, what we called gyrase, is the one that removes the supercoils created by helicase. So his is to loosen the supercoils so that helicase can continue unwinding the DNA strand. And that is what we call initiation step. And lastly, under initiation step, we shall see another enzyme which we listed here called primase, RNA primase. It brings an RNA primer. It comes and inserts here at the leading strand, which is from 3' end. It is from 5' end to 3' end, and we synthesize from 5' end to 3' end. So what we will see, RNA primer is inserted, and it is inserted by an enzyme known as RNA primase. So RNA primase inserts a primer because everything begins from something. So we cannot synthesize a new strand without a primer or without something, a precursor. So primer is a short DNA RNA strand with a few nucleotides, ranging from 5 to 50 nucleotides. So it brings a primer, and this primer is a short strand containing nucleotide bases, and he inserts it at 3' end of the leading strand. And we see synthesis happens from 5' end to 3' end. So the primer is inserted by primase enzyme at 3' end, and after inserting, this one marks the end of initiation. We are going to look at now elongation step. After putting an RNA primer, after inserting the primer, we are going to see DNA polymerase coming in to elongate. How does it elongate this? And this strand at the leading is continuous from 5' end to 3' end. And who keeps on adding nucleotides by reading the bases on the template or the DNA template strand is DNA. At the leading strand, we see the action of DNA polymerase type 3. So here we are seeing this chain elongation is being done by DNA polymerase type 3. That is in prokaryotes, and in eukaryotes, we are seeing it is delta. It is the one that synthesizes and completes the elongation of the DNA strand at the leading strand of the DNA template from 5' end to 3' end. So it keeps on adding nucleotides on the 3' end. As we are seeing the structure of the DNA, that polynucleotides of ester bonds are formed at the 3' end, at the 3 position, whereby the hydroxyl group at the number 3 is the one that allows the incoming phosphate to form a phosphodiester bond, hence completing the elongation step at the leading strand. So we are seeing the DNA polymerase type that synthesizes the continuous strand on the leading strand of the DNA is DNA polymerase type 3 in prokaryotes. In eukaryotes, it is DNA polymerase delta, which is doing the role at the leading strand. After primase have added a primer, an RNA primer, it continues from there by adding nucleotides at a 3' end to form a continuous newly synthesized DNA strand. And then on the lagging strand, while lagging, we know that DNA replication is bidirectional. It is bidirectional. One strand is leading and another one is moving behind. That is what we call bidirectional. So we are seeing this one is moving from 3' end to 5' end, and this one is moving from 3' end to this. So they are in opposite direction, and that's why you call it bidirectional. So doing DNA replication, it occurs as bidirectional movement. So at the lagging strand, still because the law is synthesis of strands of DNA occur from 5' end to 3' end. So that's why we see on the lagging strand, we cannot begin from here, because the position of synthesis should be from 5' end to 3' end. So what happens here, we are going to see the primase, RNA primase adds an RNA primer. And at the lagging strand, it adds multiple. This is an RNA primer, a primer number 2, number 3, number 4. And after adding these multiple primers, we see DNA polymerase type 1 coming in to synthesize from 5' end to 3' end. He keeps elongating he starts from the primer at 5' end and takes it at the 3' end. Begins from 5' end, 3' end, like that. And he starts creating fragments. And these fragments created at the lagging strand is what we call Okazaki fragments. Okazaki fragments. And these Okazaki fragments are the ones which are created by a DNA, which we are calling DNA polymerase type 1. So this one is done, the furring or the elongation step at the lagging strand between multiple RNA primers to form Okazaki fragments is done by DNA polymerase type 1. And that is in prokaryotes. In eukaryotes, it is alpha, which does that work. And it's the one that continues to come and does gap filling. He also does the gap filling. So he elongates he creates the Okazaki fragment, and also and this process is what we call gap filling. Gap filling because it is filling the gaps between RNA primers. And after doing that, of gap filling, it reaches a step whereby it cannot continue, where it needs help. And where it needs help, there is another enzyme that comes in to help it to continue filling the gap between Okazaki fragments. So where DNA polymerase stops, we see another enzyme known as DNA ligase. And DNA ligase is the one that comes and completes the work of gap filling. So gap filling is started by DNA polymerase type 1, then it is ended by DNA ligase, which finishes the role of filling the gap at Okazaki fragments. And after forming these Okazaki fragments, now we need to knock out these primers, because these primers were to initiate elongation at the lagging phase. So after forming the Okazaki fragments, we need to remove the primers because we no longer need them. We no longer need them. Their role was to initiate chain elongation. So after elongating the chain, we have to remove them. They no longer have the importance. So we are going to see where the primers was, they are removed. And when they are removed, they leave in the gaps. And these gaps still are filled by polymerase 1, which we see polymerase type 1 continues to fill. It knocks out the primers and continues to fill to form a continuous strand from 5' end to 3' end. And after doing that, which is by polymerase type 1, there is another type which we call the type 2 in prokaryotes. This type 2, what we are calling DNA polymerase type 2, for him, he has the exonuclease activity. And this exonuclease activity of type 2, he is going to proofread. He does the role of proofreading to see if there is a mistake. It has proofreading exonuclease activity. Exonuclease activity that after forming these strands on the lagging and the leading strand, it comes proofreading to see if adenine is with guanine, if cytosine is with. So to see complementary base pairing, Where he finds there is a problem, he comes and repairs. He does also what we call DNA repair. After he has found there is a mistake in the base pairing, he removes and repairs. Proofreading, it is done from 3' end to 5' end direction. And his DNA repair exonuclease activity is from 5' end to 3' end direction. So this is the DNA polymerase type 2. And this DNA polymerase type 2, in eukaryotes, we see the rule of proofreading is done by epsilon. Epsilon in eukaryotes does the work of proofreading in 3 to 5' end. And then repair in eukaryotes is done by beta. DNA polymerase beta is the one that does repair in eukaryotes. But in prokaryotes, we see DNA polymerase type 2 doing proofreading. He comes and proofreads from 3' end to 5' end. When he finds a mistake, he comes and starts repairing from 5' end to 3' end. He comes at Okazaki fragment, reads from 3, proofreads from 3 to 5' end, and then repairs from 5' end to 3' end. In case he finds there is non-complementary base pairing, he is able to cut out and repair. So that is the role of DNA polymerase type 2. And that marks the end of DNA elongation. That is DNA elongation. And finally, we shall see another third step, which is termination. This termination is done by TER protein. TER protein is the one that recognizes the strands where termination occurs. As the helicase is cutting, when there is the time for chain termination to occur, we see termination protein coming and binds. Termination protein. It binds and stops helicase from continuing to work. So when this one, which we are calling TER protein, what we call termination protein, he binds in front of helicase and stops him from continuing to unwind DNA strands. And when he binds, he stops helicase from continuing to unwind. And when this helicase is inhibited, it leads to what we call chain termination. It is what we call chain termination, and that will be the end of DNA replication. So we have seen how 3 steps occur: DNA initiation, elongation, and termination. And we have seen different enzymes that play a role in each step. So in summary, we can see, I can draw a table here. This table, I'm going to see the enzyme and the function. And you are going to begin with DNA, what we call DNA protein B, but let us call it helicase. And this helicase is responsible for unwinding or separating DNA strands. The one that separates DNA strands. Then we saw that as he's cutting, he creates a supercoil, which are removed by topoisomerase. So topoisomerase is what we are calling DNA gyrase, which is inhibited by fluoroquinolones, is the one responsible for loosening DNA supercoils created by helicase, so that the helicase can continue to work. So if you remove the supercoils, so it acts as the bulldozer. Then another enzyme we have seen this is ligase, DNA ligase, which does a lot of also gap filling at Okazaki fragments on the lagging strand. And this one does when DNA polymerase type 1, which also does gap filling and complete formation of Okazaki fragments at the lagging strand. Then we have seen polymerase type 2 doing what we call proofreading and repair. Proofreading in the 3 to 5 prime end, then repair from 5 prime end to 3 prime end. Then we have seen another enzyme type 2 which completes type 3, the one that completes elongation or DNA strand synthesis at both leading and the lagging strand. That is the top of 3. So for him, he completes the elongation step. Then we have seen another enzyme. One more, what we have not seen is this. These are what we call single-stranded binding proteins. And these single-stranded binding proteins are the ones that prevent reannealing. It is like after we have cut the strands separate, they need something to keep attaching them on the surface so that they do not join again. So what stabilizes the separated DNA strand is what we are calling single-stranded binding protein. Single-stranded binding protein stabilizes the separated DNA strand and prevents them from reannealing or from reassociating. So that is the function of single-stranded binding protein. So these are the summary I can make here. These points to note. Do not forget that this replication is bidirectional. And who stands in the replication fork is the helicase who unwinds, and when it's unwinding, the supercoils created are removed by topoisomerase. And don't forget they occur in 3 steps, that is initiation, elongation, and termination step. And we have seen all the enzymes involved and what they do, and we have seen also the difference in the prokaryotes and the eukaryotes. They take the same steps, but the difference we have seen, prokaryotes, the DNA polymerase is named in Roman numerals, then in eukaryotes is named in the Greek letters, alpha, we had the beta, epsilon, we had the gamma, and we had also delta. And we have seen all what they do. And we have seen that even in the prokaryotes, they have, at the origin of replication, they have a single specific site. Whereas in eukaryotes, they have multiple sites. In eukaryotes, they have multiple sites. We have also seen that we can see also in prokaryotes, it is faster. The replication speed is faster compared to eukaryotes, whereby it is short. And even the primers in the prokaryotes, they are longer, 5 to 500 nucleotide, whereas in eukaryotes, the primers are short. Thank you so much for being tuned in. Always remember to subscribe at Medical Sciences by Naphtali Muhumuza. Remember to hit the like button, share your comments, and we shall continue to grow. Thank you so much.